my name is Daniela Salgado. I'm a second-year PhD student at UC San Diego and La Jolla Institute for Immunology, and I'm in Dr. Ferhat iLab. And today I'm going to present a multiscale 3D chromatin architecture that controls lineage commitment. Our lab mainly focuses on developing methods and analyzing chromatin data to understand how the 3D genome organization affect different biological processes, such as differentiation and disease progression. But how can we do that? Currently, we have multiple next-generation sequencing technologies for characterizing chromatin organization. One of these is ChIP-seq, that it measures protein DNA interactions, and it allows us to extract DNA binding sites for specific proteins. To study 3D organization, we mainly use HiC and HiChIP. HiC mainly relies on DNA cross-linking using formaldehyde, and then the chromatin fragments are cut and ligated. From these, we can extract this kind of contact maps where each pixel represents the frequency of interaction between two regions of the genome. HiChIP mainly combines ChIP-seq and HiC, and it has an extra step where we do chromatin immunoprecipitation, and it allows us to extract loops that are specific to a particular protein. With this type of chromatin capture assays, we now know that the genome is not randomly organized. Instead, its structure is very important for defining cell types. This was illustrated by Waddington with the epigenetic landscape, where at the top of the mountain there's a stem cell, and depending on each path it follows, it can differentiate into a cell type. And these paths represent interaction between different parts of the genome and interaction with its environment. But to achieve an epigenetic landscape that is cell type specific, the genome is organized into different features at multiple scales. First, with the chromosome territories, in turn, these are organized into compartment, topological associated domains, or TADs, and loops. We believe that the loops are formed by loop extrusion, where cohesin binds to DNA and it begins extruding until it finds ETCF that is convergently oriented. But how do lineage determining transcription factors organize the 3D genome structure to support and enforce lineage choice are not yet fully understood. To answer this question, we examine the effect of loss of function of Icarus, a DNA binding protein that is important for B-cell differentiation. This protein has 4 zinc finger domains that allow DNA binding and 2 domains for dimerization. So when this process of B-cell differentiation and the transcription factors involved in it were being described, it came into realization that the sites where those factors were acting upon were very far apart from each other, and somehow they were brought into close proximity to induce developmental genes. So to understand how Icarus is bringing these sites together, we perform in situ HiC and HiChIP in combination with ChIP-seq for different histone modification and transcription factors, and also RNA-seq. And we did this in vivo wild type and Icarus mutant large PV cells, also in an inducible system where we induce the deletion of Icarus in vitro in wild type large PV cells, and in some gain of function experiments where we induce the expression of Icarus in keratinocytes, where it is not normally expressed. HiC and HiChIP reveal the expected chromatin organization into compartment, topological associated domains, and chromatin loops, and also reveal striking changes between wild type and Ankydian. Then, to examine changes of loops in a systematically way, we did this aggregate approach, where we start from a set of differential loops, and then we aggregate this by taking the average of each pixel that it's a specific genomic location. And we did this in 2 conditions, for example, condition 1 and condition 2. And in this way, we can confirm if our loops are differentially expressed in one of these conditions. This will be an example where the loops are gone in condition 2. So we did this aggregate analysis with HiC and H3K27 acetylation loops. And we confirmed that the loops that we identified as downregulated are completely gone in IKDN. Using Icarus high chip, we saw that there was a strong and very specific enrichment between the loops assembled by Icarus and the downregulated loops. Then we wanted to see if Icarus was present at these loops. So we used the chip-seq peaks and overlapped them with the loop anchors. We saw that more than 80% of these differential interactions had Icarus in at least one of its anchor. And specifically for the downregulated H3K27 acetylation, all of them had Icarus bound. Then, to examine the connection between the effect of Icarus loss on chromatin organization with gene expression, we first identified 1,200 of upregulated genes and 400 downregulated. We then associated these loops with upregulated and downregulated H3K27 acetylation loops. From this, we identified that out of these 400, 360 had a contact to its promoter in wild type, and for 80% of these genes, this contact was disrupted in IKDN. One specific example is VLK that is required for pre-VCR signaling. And here in this locus, we see that the interactions between these two clusters of enhancers that are identified in wild type are lost, as well as the enrichment for K27 acetylation, for NIVO, and for SMC. But for CTCF, it happened something interesting where we observe a loss of interactions despite the minimal changes in CTCF occupancy. And we observe very similar pattern in other loci, like the J chain and the MS4A gene cluster. Icarus loss also resulted in a disruption of structural loops. And to understand how Icarus was maintaining these type of loops, first we look at the loop anchors. We wanted to see what was bound to those regions and if that was changing. So we found that CTCF and SMC were highly enriched at the loop anchors, but CTCF was not changing. However, all these structural loops span an enhancer. And then when we look at these regions, we saw that there was a loss of H3K27 acetylation and SMC. And this suggests a very cool mechanism in which cohesin is decreased in the internal enhancers, and this also led to a decrease of cohesin in the anchors and a weakening of these loops. So this suggests that Icarus regulation of structural loops is independent of changes in CTCF bound anchors. Then we wanted to understand how Icarus was interacting with CTCF. CTCF is a DNA binding protein that promotes insulation. So in an aggregate plot, it looks like this, where there's an enrichment of interactions at both sides of this site, but there are not interactions crossing this site. When we look at enhancers that are bound by Icarus and not CTCF, we can see that they are being crossed by very long range interactions. And when CTCF is bound, we observe a reduction of this pattern, but still a lack of insulation. In contrast to enhancers, when we look at the promoters, we saw that those that have Icarus bound and not CTCF were being spanned by short range interactions that were almost gone when CTCF was bound. So this suggests that Icarus bound enhancers override to a certain extent the CTCF insulation. Taking all these together, our study demonstrates two ways in which Icarus interacts with cohesin and CTCF to organize chromatin. Icarus binding at internal enhancers is necessary for cohesin loading and also for maintaining structural loops. And Icarus is also effective at overcoming the CTCF insulating effects to promote interactions that cross the CTCF boundaries. One specific example is the IG kappa locus, where here in IKDN we can see that there's a loss of interactions between these topological associated domains that are identified in wild type. And we further confirm this with imaging, where here in IKDN we can see that these two regions from here are no longer interacting. Then, to study the dynamic changes in chromatin organization, we perform an inducible in vitro deletion of Icarus. And here we can see how the differential loops that were identified in wild type versus IKDN are progressively changing. And then we cluster these interactions to see if we can extract different clusters of interactions that were changing at different rates. And we identify these three main clusters where we have interactions that are slowly changing and also fast changing. And when we look at the enrichment of H3K27 acetylation at these anchors, we saw that a substantial fraction of loop changes precede in changes in H3K27 acetylation. Finally, to confirm Icarus' ability to promote interactions and also to organize the genome, we perform a gain-of-function experiment where we induce the expression of Icarus in Hackett cells. Here is not normally expressed. After the induction of expression, we saw multiple compartmental changes, most of them shifting towards euchromatin and some of them towards heterochromatin. When we associated the differential express genes with regulatory loops, we saw that around 60% of the upregulated genes were associated with loops that were induced after Icarus was expressed. Although Icarus didn't confer a lymphoid identity to these dermal cells, we saw that Icarus organizes some of the lymphoid-specific genes in a lymphoid-like chromatin conformation. One example is CD28 and CTLA4, where in mouse privy cells, it is located into a permissive site of the chromatin that is highly organized by Icarus. And when we look at Hackett cells, we saw that Icarus binds and also promote interactions between distal enhancers that are located in this region where there's a flip from weak heterochromatin to euchromatin. And this confirms that Icarus has the ability to promote interactions with distal enhancers and promoters, and also to confer a euchromatin location to the regions that have induced loops. In conclusion, here I show how Icarus assembled a 3D genome organization that presages lymphoid lineage gene expression, and cohesin loading at Icarus-bound enhancers sustain the CTCF structural loops, and the enhancers that have Icarus bound have the ability to override CTCF insulation to assemble lineage-specific 3D organization. And I would like to thank first my supervisor, Dr. Ferhat Ay, and Surya, that is a co-author of this paper, and all my lab and collaborators. Thank you.
Thanks, Daniela, for a beautiful talk. We have time for a few questions.
So what you observe for Icarus is that playing this role in 3D genome organization.
Yes.
Is that specific to the lymphoid lineage, or do you think that there are similar types of factors, or maybe Icarus would be your role in other?
Yeah, there are other factors. One good example is CDF1, that it induces these chromatin changes for lymphoid development. Also, another factor is myodine, that it performs these changes in chromatin in muscle cells. So yeah, there are a lot of factors that can have this ability.